The autonomic nervous system controls internal body processes such as the following: blood pressure, heart and breathing rates, body temperature, digestion, metabolism, which affects body weight, the balance of water and electrolytes, such as sodium and calcium, the production of body fluids, including saliva, sweat, and tears, and urination, defecation, and sexual response. All of these occur autonomously, meaning without conscious effort. So the autonomic nervous system is sometimes called the involuntary nervous system, or because these involve the viscera, or the organs, the general visceral motor system. The autonomic nervous system is part of the motor, or efferent division of the PNS. The somatic nervous system and the autonomic nervous system both consist of motor neurons, but there are three key differences between them: the effectors or targets that the motor neurons innervate, the nature of the efferent pathways and whether or not ganglia are present, and the response of target organs to neurotransmitters. Motor neurons of the somatic nervous system innervate skeletal muscles. The cell bodies lie in the ventral horn of the spinal cord. The cell bodies lie in the ventral horn of the spinal cord, which is in the CNS. A single thick myelinated axon travels out of the CNS and into the PNS via the spinal or cranial nerves and continues uninterrupted to its target organ, skeletal muscle. Nerve terminals of somatic motor neurons release acetylcholine, which always stimulates skeletal muscles to contract. Motor neurons of the autonomic nervous system innervate cardiac muscle, smooth muscle, and glands. The ANS uses a 2-neuron chain to reach its effectors, kind of like having to take one bus to a station, disembarking, and then hopping onto a second bus to your destination. The first leg of the journey starts in the CNS, where preganglionic neuron cell bodies reside in the lateral horn of the spinal cord or in the brainstem. You can see that in the last slide. The lightly myelinated preganglionic axons exit the CNS via the spinal and cranial nerves and extend to the autonomic ganglia in the PNS, where they synapse on the cell bodies of second-order neurons and release acetylcholine, which is stimulatory at these synapses. The non-myelinated axons of the second-order neurons travel from the ganglia to their target effector organs. The exception is the adrenal medulla, which is stimulated by a preganglionic, i.e., first-order neuron that releases acetylcholine. The cells of the adrenal medulla have the same embryonic origin as neural tissue, and in fact, function as modified postganglionic neurons. Instead of releasing neurotransmitter directly at the synapse within an effector tissue, the secretory products of the adrenal medulla are picked up by the blood and travel throughout the body to all of the effector tissues of the sympathetic system. Autonomic postganglionic fibers release two neurotransmitters, norepinephrine, secreted by most sympathetic fibers, and acetylcholine, secreted by parasympathetic fibers. Depending on the neurotransmitter and the type of receptors on the target organ, the effect may be excitatory or inhibitory. So the ANS differs from the SNS in that it can stimulate or inhibit its effectors. The ANS is composed of two anatomically and functionally distinct divisions, the sympathetic system and the parasympathetic system. Both systems are tonically active. In other words, they provide some degree of nervous input to a given tissue at all times. Therefore, the frequency of action potentials of neurons in both systems can either increase or decrease. As a result, tissue activity may be either enhanced or inhibited. This characteristic of the autonomic nervous system improves its ability to more precisely regulate a tissue's function. Without tonic activity, nervous input to a tissue could only increase. Many tissues are innervated by both systems. Because the sympathetic system and the parasympathetic system typically have opposing effects on a given tissue, increasing the activity of one system while simultaneously decreasing the activity of the other results in a very rapid and precise control of a tissue's function. This process is sometimes referred to as a dynamic antagonistic interaction. One division usually predominates, but in a few cases, the two divisions have a cooperative effect. We'll see an example of this later in the lecture. All of the changes in organ and tissue function induced by the sympathetic system work together to support strenuous physical activity, and the changes induced by the parasympathetic system are appropriate for when the body is resting. The parasympathetic system decreases heart rate, which helps to conserve energy under resting conditions. The parasympathetic system decreases heart rate, which helps to conserve energy under resting conditions. Salivary secretion is enhanced to facilitate the swallowing of food. Gastric motility and secretion are stimulated to begin the processing of ingested food. Intestinal motility and secretion are also stimulated to continue the processing and to facilitate the absorption of these nutrients. Both exocrine and endocrine secretion from the pancreas is promoted. Enzymes released from the exocrine glands of the pancreas contribute to the chemical breakdown of the food in the intestine, and insulin released from the pancreatic islets promotes the storage of nutrient molecules within the tissues once they are absorbed into the body. Another bodily maintenance type of function caused by the parasympathetic system is contraction of the urinary bladder, which results in urination. Finally, the eye is adjusted such that the pupil contracts and the lens adapts for near vision. The fight or flight reaction elicited by the sympathetic system is essentially a whole body response. Changes in organ and tissue function throughout the body are coordinated so that there's an increase in the delivery of well-oxygenated, nutrient-rich blood to the working skeletal muscles. Both heart rate and myocardial contractility are increased so that the heart pumps more blood per minute. Sympathetic stimulation of vascular smooth muscle causes widespread vasoconstriction, particularly in the organs of the gastrointestinal system and in the kidneys. This vasoconstriction serves to redirect or redistribute the blood away from these metabolically inactive tissues and towards the contracting muscles. Bronchodilation in the lungs facilitates the movement of air in and out of the lungs so that the uptake of oxygen from the atmosphere and the elimination of carbon dioxide from the body are maximized. An enhanced rate of glycogenolysis, meaning the breakdown of glycogen into its component glucose molecules, and gluconeogenesis, the formation of new glucose from non-carbohydrate sources in the liver, increases the concentration of glucose molecules in the blood. This is necessary for the brain, as glucose is the only nutrient molecule that it can utilize to form metabolic energy. An enhanced rate of lipolysis in adipose tissue increases the concentration of fatty acid molecules in the blood. Skeletal muscles then utilize these fatty acids to form metabolic energy for contraction. Generalized sweating elicited by the sympathetic system enables the individual to thermoregulate during these conditions of increased physical activity and heat production. Finally, the eye is adjusted such that the pupil dilates, letting more light in toward the retina, and the lens adapts for distance vision. In contrast to the sympathetic division's dominance of blood vessels, parasympathetic effects normally dominate the heart and the smooth muscle of digestive and urinary tract organs. These organs exhibit parasympathetic tone. This division slows the heart and dictates the normal activity levels of the digestive and urinary tracts. However, the sympathetic division can override these parasympathetic effects during times of stress. Drugs that block parasympathetic responses increase heart rate and cause fecal and urinary retention. Parasympathetic fibers activate most glands except for the adrenal glands and sweat glands of the skin. The adrenal medulla, sweat glands and erector pili muscles of the skin, the kidneys, and most blood vessels receive only sympathetic fibers. The sympathetic division mediates reflexes that regulate body temperature. For example, applying heat to the skin causes blood vessels in that area to dilate. When body temperature rises, sympathetic nerves dilate the skin's blood vessels, allowing heat to escape, and activate sweat glands to help cool the body. When body temperature falls, skin blood vessels constrict, preventing heat loss. Sympathetic impulses stimulate the kidneys to release renin, an enzyme that causes the formation of potent blood pressure increasing hormones, the so-called renin-angiotensin system. This system is a hormone system that regulates blood pressure and fluid and electrolyte balance, as well as systemic vascular resistance. Through both neural stimulation and release of adrenal medullary hormones, the sympathetic nervous system promotes a number of metabolic effects not reversed by parasympathetic activity. For example, it increases the metabolic rate of body cells, raises blood glucose levels, and mobilizes fats for use as fuels. Finally, the medullary hormones also cause skeletal muscle to contract more strongly and quickly. In the parasympathetic division, one preganglionic neuron synapses with one, or at most a few, postganglionic neurons. Additionally, all parasympathetic fibers release acetylcholine, which is quickly destroyed or hydrolyzed by acetylcholinesterase. Consequently, the parasympathetic division exerts short-lived, highly localized control over its effectors. In contrast, in the sympathetic division, preganglionic axons branch profusely as they enter the sympathetic trunk, and they synapse with postganglionic neurons at several levels, as I mentioned earlier. As a result, when the sympathetic division is activated, it responds in a diffuse and highly interconnected way. Nevertheless, parts of the sympathetic nervous system can be activated individually. For example, just because your eye pupils dilate in dim light doesn't necessarily mean that your heart rate also speeds up. Sympathetic activation produces much longer-lasting effects than parasympathetic activation. Adrenal medullary cells secrete epinephrine and norepinephrine into the blood when the sympathetic division is mobilized. These hormones reinforce and prolong the effects of the sympathetic nervous system. These effects continue for several minutes until the liver destroys the hormones. In short, sympathetic nerve impulses act only briefly, but the hormonal effects they provoke last a long time. The widespread and prolonged effect of sympathetic activation explains why we need to come down after an extremely stressful experience. Although the autonomic nervous system is not usually considered to be under voluntary control, there are 3 levels of control over the ANS by specific centers in the brainstem and spinal cord, the hypothalamus, and the cerebral cortex. At the bottom of this hierarchy is the spinal cord, which has reflexes for urination, defecation, erection, and ejaculation. These are involuntary autonomic reflexes but are subject to conscious control and can be overridden by the brain. A good example of this is micturition, a fancy word for peeing, which occurs involuntarily in infants and young children until the age of 3 to 5 years, after which it is regulated voluntarily. The brainstem reticular formation is a loose collection of neuron cell bodies in the pons, medulla, and midbrain in an area of mostly white matter. It is shown by the red region in the diagram at the bottom of the slide. The reticular formation exerts the most direct influence over autonomic functions. For example, certain motor centers in the ventrolateral medulla, cardiac and vasomotor centers, reflexively regulate heart rate and blood vessel diameter. Other medullary regions oversee gastrointestinal activities. Midbrain centers, namely the oculomotor nuclei, control the muscles concerned with pupil diameter and lens focus. The hypothalamus is the main integration center of the autonomic nervous system. It's the boss. In general, anterior hypothalamic regions direct parasympathetic functions, and posterior areas direct sympathetic functions. Hypothalamic centers exert their effects both directly and indirectly via relays through the reticular formation, which in turn influences the preganglionic motor neurons in the brainstem and spinal cord. The hypothalamus, acting through the ANS, coordinates heart activity, blood pressure, body temperature, water balance, and endocrine activity. The hypothalamus also mediates our reactions to fear and other emotions via its associations with the limbic system, the part of the brain involved in our behavioral and emotional responses, especially those we need for survival, feeding, reproduction, and caring for our young. You can find the structures of the limbic system buried deep within the brain, underneath the cerebral cortex and above the brainstem. The limbic lobe includes structures that are involved in emotional responses, as well as structures that contribute to memory function. It includes the hippocampus, the amygdala, and the cingulate gyrus. The limbic lobe has strong connections with the hypothalamus and influences the state of its activity on the basis of emotional state. For example, when you're anxious or scared, the amygdala will send signals to the hypothalamus along the medial forebrain bundle that will stimulate the sympathetic fight or flight response. The hypothalamus will also stimulate the release of stress hormones through its control of the endocrine system in response to amygdala input. In this way, the hypothalamus serves as the keystone or the gatekeeper of the emotional and visceral brain. Through its centers, emotions influence ANS function and behavior. Originally, scientists believed that the ANS was not subject to voluntary controls. However, we've all had occasions when just remembering a frightening event made our heart race via the sympathetic response, or the thought of a favorite food, for example, made our mouth water via a parasympathetic response. The cerebral cortex, including the insular, orbital, and medial prefrontal cortical areas, control smooth muscle and the glandular physiological processes through the sympathetic and parasympathetic nervous system. These inputs converge on the hypothalamus through its connections to the limbic system. At the review session, remind me to tell you about how my son's epilepsy relates to this. Finally, biofeedback or meditation attempts to gain awareness of physiological conditions within the body with the goal of consciously influencing them. Biofeedback training allows some people to control migraines and to manage stress. Smooth muscle contractility regulates the function of the vascular, respiratory, gastrointestinal, and genitourinary systems. Contractile malfunction or deficient control of smooth muscle activity in these systems leads to a host of clinical disorders. The sympathetic nervous system becomes overactive in a number of diseases. For example, the SNS plays a key role in regulating arterial blood pressure in humans. A boost of sympathetic signaling by long-term stress enhances tone in smooth muscles and raises the blood pressure, which can cause hypertension. In this case, the heart must work harder and artery walls are subjected to increased wear and tear. This disorder can be treated with beta-adrenergic receptor blocking drugs, also known as beta blockers.
I'm an alpha male on beta blockers.
Similarly, ulcers can be caused by increased sympathetic vasoconstrictor response to stress, which decreases blood flow to the stomach wall.